I'm Mahitha, and we're from the FRI stream Virtual Cures.
And I'm Raina. Our project focuses on discovering novel inhibitors for D-alanine-dealanine ligase in Yersinia pestis, the bacteria that caused the bubonic plague and is a bioterrorism threat today. Current treatments are only effective in early stages and face potential antibiotic resistance, which is why we chose this bacteria. Our specific protein target is D-alanine-dealanine ligase, which is critical for bacterial cell wall formation, has no human homologue, is cytosolic, and has high druggability. Our objective is to identify small molecule novel inhibitors against this protein.
We started with virtual screening, which predicts how well compounds bind to the active site of the protein. We used Gold as our virtual screening software and conducted two separate dockings at different parts of the active site, the dealanine side, which I did, and the ADP side, which Raina did. We first docked positive and negative controls to assess Gold's functionality for molecular docking. Control docking for the ADP side showed high scores for the structural analogs of the natural substrate, confirming Gold's functionality. The control docking for the dealanine side of the active site yielded similar results.
Next, we screened over 100,000 compounds from the CB-Zinc pH7 library. For both docking strategies, we used 3 filtering runs to find top-scoring compounds that follow Lipinski's rule of 5 for drug likeness. For the dealanine side, the top 10 scores range from 84 to 92, and Zinc 400 was chosen for lab testing. For the ADP side, the top 10 scores ranged from 97 to 101, and Zinc 443 was chosen. Primal images confirmed good fit in their respective active sites.
To create YP protein, first its plasmid was transformed into E. coli BL21DE3 cells optimized for protein expression. After expression, protein purification was performed with nickel anti-chromatography, and an SCS-pH gel was used to verify the purity and size consistency with the target protein. According to the gel, wells 5, 6, and 7 correspond to the protein elutions obtained, and the strongest span was around 34 kilodaltons, which matches up with our target's molecular weight of 33.15 kilodalton.
With the protein elutions in the ordered compounds, we ran differential scanning fluorometry, which is a thermal melt assay that tracks protein unfolding. This was run without the compounds to check for denaturation, and then with the compounds to see if there is a shift in melting temperature, the inflection point of the graph, which would indicate binding. This is from one run of the DSF binding assay, which showed a rightward shift in melting temperature with both Zinc 400 and Zinc 443, suggesting potential binding to the protein.
In conclusion, virtual screening identified top compounds for both areas of the active site, dealanine and ADP, and a preliminary DSF binding assay reveals that they could potentially bind to the protein. Next, we will conduct more trials of DSF in an ATP-based enzyme assay to verify the top compounds as inhibitors. The discovery of novel inhibitors will enable further development of these molecules as potential treatments for the plague.
Thank you for listening to our presentation. We would like to thank Dr. Josh Beckham for his guidance and mentorship, our mentors, and our peers in Virtual Cures for their support.